Hello students, let me talk about antigens and immunogens. Introduction. Substances that can be recognized by the immunoglobulin receptor of B cell or by the T cell receptors when complex with MHC are called antigens. Antigenicity is the ability to combine specifically with the final products of the immune response, that is, antibodies and or cell surface receptors. Immunogenicity is the ability to induce a humoral or cell-mediated immune response. Antigens can be more appropriately called an immunogen. Although all molecules that have the property of immunogenicity also have the property of antigenicity, the reverse is not true. Some small molecules called haptens are antigenic but incapable by themselves of inducing a specific immune response. Factors that influence immunogenicity. The immune system actually recognizes particular macromolecules as an infectious agent, generally either proteins or polysaccharide. Proteins are the most potent immunogens, with polysaccharide ranking second. Lipids and nucleic acid of an infectious agent generally do not serve as immunogens unless they are complex with a protein or polysaccharide. Immunologists tend to use proteins or polysaccharide as immunogens in most experimental studies of humoral immunity. For cell-mediated immunity, only proteins and some lipids and glycolipids serve as immunogens. Immunogenicity is not an intrinsic property of an antigen, but rather depends on a number of properties of the particular biological system that the antigen encounters. The nature of the immunogen contributes to immunogenicity. Immunogenicity is determined by four properties of the immunogens: its foreignness, molecular size, chemical composition and complexity, and ability to be processed and presented with an MHC molecule on the surface of an antigen-presenting cell or altered cell. Foreignness. In order to elicit an immune response, a molecule must be recognized as non-self by the biological system. When an antigen is introduced into an organism, the degree of its immunogenicity depends on the degree of its foreignness. The greater the phylogenetic distance between the two species, the greater the structural and therefore the antigenic disparity between them. Molecular size. There is a correlation between the size of a macromolecule and its immunogenicity. The most active immunogens tend to have a molecular mass of 1 lakh Dalton. Generally, substances with a molecular mass less than 5,000 or 10,000 Dalton are poor immunogens, although a few substances with a molecular mass less than 1,000 Dalton have proven to be immunogenic. Chemical composition and heterogeneity. Size and foreignness are not by themselves sufficient to make a molecule immunogenic. Other properties are needed as well. For example, synthetic homopolymers, polymers composed of a single amino acid or sugar, tends to lack immunogenicity regardless of their size. Copolymers composed of different amino acids or sugars are usually more immunogenic than homopolymers of their constituents. All four levels of protein organization, primary, secondary, tertiary, and quaternary, contribute to the structural complexity of the protein and hence affect its immunogenicity. Susceptibility to antigen processing and presentation. Large insoluble macromolecules generally are more immunogenic than small soluble ones because the larger molecules are more readily phagocytosed and processed. Macromolecules that cannot be degraded and presented with MHC molecules are poor immunogens. For example, Degradative enzymes within antigen-presenting cells can degrade only proteins containing L amino acids. Polymers of D amino acids cannot be processed and thus are poor immunogens. The biological system contributes to immunogenicity. Even if a macromolecule has a property that contributes to immunogenicity, its ability to induce an immune response will depend on certain properties of the biological system that the antigen encounters. These properties include the genotype of the recipient, the dose and route of antigen administration, and the administration of the substance called adjuvant that increase immune response. Genotype of the recipient animal. The genetic constitution or genotype of an immunized animal influence the type of immune response that the animal manifests as well as the degree of the response. For example, Hugh McDavid showed that two different inbred strains of mice responded very differently to a synthetic polypeptide immunogen. After exposure to the immunogen, one strain produced high level of serum antibody, whereas the other strain produced low levels. When the two strains were crossed, the F1 generation showed an immediate response to the immunogen. By back cross analysis, the gene controlling immune responsiveness was mapped to a subregion of the measure MHC. MHC gene products which function to present processed antigen to T cells play a central role in determining the degree to which an animal responds to an immunogen. The response of an animal to an antigen is also influenced by the genes that encode B cell and T cell receptors and by genes that encode various proteins involved in immune regulatory mechanisms. Genetic variability in all of these genes affects the immunogenicity of a given macromolecule in different animals. Immunogen doses and route of administration. Each experimental immunogen exhibits a particular dose-response curve which is determined by measuring the immune response to different doses and different administration routes. An antibody response is measured by determining the level of antibody present in the serum of immunized animals. Evaluating T cell response is less simple but may be determined by evaluating the increase in the number of T cells bearing TCRs that recognize the immunogens. Some combination of optimal doses and route of administration will induce a peak immune response in a given animal. A single dose of most experimental immunogens will not induce a strong response. Rather, repeated administration over a period of weeks is usually required. Such repeated administration or boosters increases the clonal proliferation of antigen-specific T cells or B cells and thus increase the lymphocyte populations specific for the immunogen. Experimental immunogens are generally administered in the following routes: intravenous or IV into a vein, intradermal or ID into the skin, subcutaneous or SC beneath the skin, intramuscular or IM into a muscle, intraperitoneal or IP into the peritoneal cavity. The administration route strongly influences which immune organs and cell population will be involved in the response. Antigen administered intravenously is carried first to the spleen, where antigen administered subcutaneously moves first to the local lymph nodes. Differences in the lymphoid cells that populate these organs may be reflected in the subsequent immune response. Adjuvants. Adjuvants are substances that, when mixed with an antigen and injected with it, enhance the immunogenicity of that antigen. Adjuvants are often used to boost the immune response when an antigen has low immunogenicity or when only small amount of an antigen are available. Adjuvants exert one or more of the following effects. Antigen persistence is prolonged. Co-stimulatory signals are enhanced. Local inflammation is increased. The non-specific proliferation of lymphocyte is stimulated. Some of the common adjuvants are aluminum potassium sulfate or alum, Freund's incomplete adjuvant, Freund's complete adjuvants, etc. Epitopes. Immune cells do not interact with or recognize an entire immunogen molecule. Instead, lymphocytes recognize discrete sites on the macromolecules called epitope or antigenic determinants. Epitopes are the immunogenically active regions of an immunogen that binds to antigen-specific membrane receptors on lymphocytes or to secreted antibodies. B and T cell recognize different epitopes on the same antigenic molecule. For example, when mice were immunized with glucagon, a small human hormone of 29 amino acids, antibodies were elicited to epitopes in the amino terminal portion, whereas the T cell responded only to epitopes in the carboxyl terminal portion. Lymphocytes may interact with a complex antigen on several levels of antigen structure. An epitope on a protein antigen may involve elements of the primary, secondary, tertiary, and even quaternary structure of the protein. In polysaccharides, branch chains are commonly present and multiple branches may contribute to the conformation of epitopes. The recognition of antigens by T cells and B cell is fundamentally different. B cell recognize soluble antigen when it binds to different membrane-bound antibody. B cells recognize soluble antigen when it binds to their membrane-bound antibody. Because B cell bind antigen that is free in solution, the epitopes they recognize tend to be highly accessible sites on the exposed surface of the immunogen. Most T cells recognize only peptides combined with MHC molecule on the surface of antigen-presenting cells and altered self cells. T cell epitopes cannot be considered apart from their associated MHC molecules. T cells tends to recognize internal peptides that are exposed by processing within antigen-presenting cells or altered self cells. Haptens. Haptens are small organic molecules that are antigenic but not immunogenic. Chemical coupling of a hapten to a large protein called a carrier yield an immunogenic hapten carrier conjugates. Animals immunized with such a conjugate produce antibodies specific for the hapten determinant, the unaltered epitope on the carrier protein, and new epitopes formed by combined part of both the hapten and the carrier. By itself, a hapten cannot function as an immunogenic epitope, but when multiple molecules on a single hapten are coupled to a carrier protein or non-immunogenic homopolymer, the hapten become accessible to the immune system and can function as an immunogen. Many biologically important substances, including drugs, peptide hormones, and steroid hormones, can function as haptens. Conjugates of these haptens with large protein carrier can be used to produce hapten-specific antibodies. Lipids as antigens. Appropriately presented lipoidal antigen can induce B and T cell response. For the stimulation of B cell response, lipids are used as hapten and attached to suitable carrier molecules such as the protein keyhole limpet hemocyanin or bovine serum albumin. Using this approach, antibodies have been raised against a wide variety of lipid molecules including steroids, complex fatty acid derivatives, and fat-soluble vitamins such as vitamin E. For example, a determination of the level of a complex group of lipids known as leukotrienes can be useful in evaluating asthma patients. This becomes possible by developing antibody against leukotrienes. Lipoidal compounds such as glycolipids and some phospholipids can be recognized by T cell receptors when presented as complex with molecules that are very much like MSC molecules. These lipid-presenting molecules are members of the CD1 family and are close structural relatives of class 1 MSC molecules. Conclusion. In conclusion, I would like to state that antigens are substances that can be recognized by the receptors on B cells and T cells. Any antigen that can elicit an immune response are immunogens. Immunogenicity or the ability to induce immune response depends on the nature of the immunogens and also on the biological system in which the immunogens are introduced. Immune cells recognize discrete regions on the antigens called epitopes or antigenic determinants. Therefore, antigens can have many epitopes recognized by different B and T cells. There are other small organic molecules called haptens that are antigenic but not immunogenic. They can be conjugated with proteins to make them immunogenic. Many biologically important substances can function as hapten, and when they are conjugated with proteins, a large number of hapten-specific antibodies could be produced which have varied applications.